All right, good morning, everybody. Any questions before I get going? All right, so today's stuff, this week's stuff, we want to think a little bit about how we go about seeing some of these organisms in the first place. There are all sorts of different tools that we might potentially employ. We've already hinted, I guess, about the lab, the littlest bit. I know it's starting this week for everybody. We'll look at organisms, bacteria, with a compound light microscope, and it allows us to see the bacteria as sort of little specks of cells. We will use different types of stains. Really, for us, we're just going to do a gram stain, but different types of staining procedures that allows for the organisms to be visualized considerably better than if they were just put on the slide. They kind of look like hollow little cells, ghost cells, if they haven't been stained up. So that's how we go about it, but we could dive deeper and look at them more magnified with electron microscopes. There are other techniques that might be employed as well. There's also a handful of different stains that give you some other details, features that a cell might possess or maybe not. And that might be definitely of importance when we're trying to weed through the plethora of organisms that we're trying to figure out who are we really dealing with, right? So a lot of ways, and there are numerous techniques as we move through here that I'm going to kind of gloss over the littlest bit and spend a little more time just looking at some of the images of what those respective microscopes are going to allow us to see. With that said, the biology department has a handful of these microscopes that we're going to be looking at in this lecture. But most of them we really think about more kind of research oriented. It's not something that just typical teaching labs spend their time looking at things like that. And it's also something that's really pretty complex, right? We'll touch on some of that because there are a few courses that do get an opportunity to kind of dive into that. And then we'll discuss that as we move through here the littlest bit. So as we get going, and there are lots of images showing different types of microbes as we progress along. So this gives us opportunities to really kind of get our first hint at what we're looking at. And in a clinical situation, oftentimes samples will be collected, they get brought back to the lab, and there are numerous ways that this stuff might be occurring. We're not going to dive into that right now, but whatever specimen was collected, it can be brought into the lab, and typically the first thing that we're going to think about is staining up this stuff to see what sorts of potentials we might be dealing with. There are other opportunities where maybe it immediately gets cultivated. If it's a blood sample, something like that, thinking about maybe some sepsis, we would cultivate that first. And then after it's had, time to incubate, that's when we think about the potential to see maybe what's in there, all right? But lots of samples, the first thing that's going to happen is some staining. And then based off of the staining characteristics that we might observe, that's going to tell us what sorts of media we might be putting these specimens on to further allow us to weed through it and differentiate really who we're looking at. All right. So that's kind of the idea, sort of a first step with many of these techniques. So the bright field microscopy, as we see right here, this is the compound microscope like you'll be working with in the lab. All right. This image has some more stuff, all right, than just bacterial cells. So these big rectangular or kind of boxcar-like train car is how these guys get described. And they're sort of, some of them linked together. This is anthrax, Bacillus anthracis, all right, a type of long rod or bacillus bacterium, and they're purple colored. That tells me a little something about these organisms when I see that. All right, this is a gram stain, and we can see cells, bacterial cells, that might appear purple, purple-ish, or pink, reddish pink, right? Dependent really on the characteristics of that bacteria's cell wall, right? And we'll go into some more details with that a little bit later. But bacteria, typically, bacteria could have very thick cell walls with lots of sugar protein cross-linkings. We call that peptidoglycan, and we call those cells gram-positive. Or it can be a very thin layer of cell wall material, peptidoglycan, and those are gram-negatives. And based off of that, what their cell wall is like can clue us into some details about the bacteria, and it also can clue us into types of media or petri dishes that we might want to use to separate out some of these things. All right. So along with all of the bacillus that we're seeing here are some technically gigantic cells compared to the bacteria. These are all white blood cells. So with that, and this is coming from cerebral spinal fluid, we showed earlier on that image and the little vial that had maybe some spinal fluid in, and it looked nice and clear. In this instance, my gut tells me it wouldn't look clear. It'd be pretty turbid, right? Full of white blood cells, as we see here, and the bacteria. And when we stain up that CSF, we're able to visualize all of that, all right? So with that said, there are numerous types of white blood cells in here, and a lot of them right in here we will call neutrophils. These are some of not some, they are the most dominant type of white blood cell when we think about in our circulation. And they truly are some of the first responders when it comes to these types of infections, where they can recognize and respond. If we move into the tissues, macrophages would be more of the first responders to kind of seek and stimulate others to come in and respond. All right. So neutrophils have the potential to move out of the bloodstream and get into tissues or into the spinal fluid, as we see here, and to potentially do their job of destroying. All right. That's really what neutrophils, their role is, kind of assassins or maybe kamikazes, because they kill themselves in the process of destroying whoever they're coming in contact with. All right. And then these others where you can see the nucleus is gigantic in a handful, maybe 4 cells. These are all types of lymphocytes, a B cell or a T cell, and we don't really, just by staining, you can't really tell with that. But there are other white blood cells we might be able to differentiate based off of some staining. And we'll think about that stuff later on. All right. So this with the light microscopy, the background, the light is shining through, the cells have been stained up, and you get pretty good resolution. I'm assuming this is under oil immersion, so 1,000-time magnification. Doesn't tell us that stuff, but just looking at these cells, it's got to be zoomed in like that, all right? Next door is a different type of microscopy, and it gives pretty good contrast. This is what we call dark field microscopy, and it's the opposite, all right? So the background is darkened out, and the cells, the light is basically kind of shining right through them. So it's sort of glow in the dark sorts of stuff, but they're not really glowing, all right? There are some other staining techniques, fluorescence microscopy, where those cells truly would be glowing, but that's a completely different type of procedure and a different type of microscope to see it, all right? So this is a type of spirocate Borrelia burgdorferi, and this is the culprit for Lyme disease. So that's what we're seeing there, and I assume also a giant white blood cell, but with that type of microscopy, can't tell what it is, all right? So there are definitely some details in here that are important when we think about how microscopes are functioning, but we're going to start showing some of this stuff and comparing like compound or light microscope and electron microscopes. And I don't want you to get bogged down with all of that, the science behind what's happening with these, the light or the electrons. Just recognize there's differences, all right? So with this, as we look at the wavelength from one peak to the next, all right, is a wavelength, and we can measure these close together or farther apart and really think of it as types of energy. The closer they are, the higher the frequency, more energy within there, and the farther apart, less, all right? The visible spectrum is a specific wavelength of light that we can perceive. We'll see a bit of that in a second, all right? So how high that peak is the amplitude, all right? And we can measure this in over a unit of time, potentially in a second, to get these ideas of frequencies of our wavelengths. So you're going to look at all sorts of types of media in the lab. If it's a single one, we call it a medium, but many media, all right? So they're going to be in plastic Petri dishes, all right? And solidified with some agar that allows it to stay solid when we want to cook it up at like 37 degrees Celsius, all right? That's important, all right? These Petri dishes can hold all sorts of different types of media, and you'll see a good chunk of them as you move through, all right? The plastic is transparent, allowing you to visualize what's going on that plate, potentially without ever even removing the lid, all right? Oftentimes, the lids will be pulled off and you'll use your inoculating loop and take something from the plate, put it on to a tube or something else or to a different plate. But you can also just look at it and see, is there growth or not? Typically with bacteria, we're looking down through the lid at the surface of the plate. If we were looking at fungus, some mold growing on a Petri dish, we would look at the top side of it, and we will also look at the bottom side of it, because oftentimes the fungus looks really different on the bottom side. And that is something that might clue us into different types of fungi that we might be dealing with, all right? But with bacteria, it's really just the top, all right? And there's all sorts of different details. You'll do a little of this in the lab pretty soon when we start thinking about how bacteria grow on different types of media. But on a solid surface like an agar plate, you get colonies of bacteria. They may be very close to the surface or raised up. They may look nice and concentric or very jagged, kind of root-like process. Numerous ways that the bacteria can grow. Some of them look very dull, others nice and shiny, and some of those things can clue you into some potential details about it. And we'll touch on some of that as we move through. So this next bit, transparent. This is a slice of meteorite opaque, right? You can't see through that. So there's numerous things we need to kind of take into account when we're looking at a specimen on a slide through the microscope. All right. We really kind of just take it for granted and just slap a slide on the stage of the microscope and look at that respective image and go about our business. All right. But there's more to it really when we think about it. So here's a chunk of glass, microscope slide. All right. Is really what we could be imagining this as. It looks pretty thick to me, but that's what I want it to be. So some light, and as it hits that material, whatever it might be, the light potentially bends, typically does, all right? So it gets refracted here. The angle of it changes, all right? And then as it's moving in, refracted, and then reflected ray as it comes out, a little bit moved, all right? The background here, this is opaque, right? And then our transparent stuff is our little ray of light is moving through there, all right? So this is another image as the light is moving from one medium to another, right? That becomes important. Like from that microscope, from the light source, typically down at the bottom, there are other microscopes like dissecting microscopes that might have light at the bottom or up at the top, depending on what you want. But with what we do looking at bacteria, it's at the bottom and it shines up through the stage. And as that's happening, as the light's moving from air into glass and then from glass into the lens, there's definitely going to be some of that light that's not captured by our lenses of the microscope. We're going to get some of it reflected away as it gets refracted as it hits the glass. So we lose some of that. And that impacts the resolution, our ability to really see these images and to see them as separate when they're close together. It has an impact, right? When we want to see something like, flipping it back to that bacillus that we started with, and I said, high power, 1000 magnification, we call that oil immersion. And we put oil on the slide and it touches the lens to try to maximize the amount of light that we're getting through there so we don't lose some of it in the process, all right? We'll see some of that here in just a second. So this is another image, another example of how what we are seeing can be altered with the refractive indices of air versus the water. It looks bent, right? This metal rod is nice and straight, I assume, all right? But when it goes into the water, it looks as if it has bent, all right? Because the refractive index of air and water are different. And that gives us, well, the ability for things to look distorted. That's an issue. When we're trying to see things through the microscope, we don't really want them to be distorted, all right? And that's like what I was just saying with the little droplet of oil on the slide helps to prevent this, all right? So this is, refractive indexes looking at how basically kind of the thickness as we think of that as some substance or light is moving from one thing into the next. There, lots of the immersion oil, the little droppers that we will be putting oil onto the slides, lots of them are little plastic squeeze bottle droppers, all right? And a little drop of oil goes onto the slide. All fine and good, all right? The old school immersion oil, and there are definitely some bottles that we still have in the labs. They're little glass bottles with droppers on the inside, but it's not like a little bulb that you depress and get liquid in it and drop stuff out. It's a glass rod in this dropper bottle. And the glass rod, when you pull it out of there, has a little drop of oil on it, and you can touch it onto the slide and put it back in the bottle, all right? What I found pretty impressive my first go of microbiology as an undergraduate was seeing those dropper bottles, and it's almost like magic. I felt like you can see the dropper bottle and you can see the oil inside of the dropper bottle, right? You can see kind of a little line, right? That's where the oil basically is at, but really you're not seeing the oil that much, right? The dropper itself, you can see the glass rod, but as soon as it touches the oil that's in that bottle, you don't see the glass rod anymore. It's invisible because the glass and the immersion oil have the same refractive index. Magic. It's pretty impressive, all right? If you happen to be in the lab and see some of the bottles, glass bottles that are for the immersion oil, just take a look. You will see what I'm talking about, right? And as soon as you pull it out of there, you see the entire little glass rod. But once it goes in, anything that's hitting the oil, it's invisible, all right? That's pretty cool, all right? I feel like that's enough. We should just leave it at that. All right. So there's different ways that we can see these things through lenses. Glass that's been ground down to give us the opportunity to focus that light at a specific area, a focal point, all right? First up, well, lenses here, but this first one is a prism. And you've all probably seen some prisms as, natural light hits the prism. It allows it to be separated out into the visible spectrum, a rainbow, right? You can see all of that as it's moving through there. So the light as it moves through here, this prism has a high refractive index as we're moving through, and the refracted light, as it blows it out into its spectrum, lower, all right? We don't need to get bogged down or worry about any of that. Moving through, there's different types of lenses. Convex, concave, it kind of makes me think, although these are lenses and we're seeing through there or light's passing through there, it makes me think of like funhouse mirrors where they're curved one way or bent in the other way, convex or concave, and you look very different, right? Super tall, super short, one way or the other, all right? So as we look at this, the lenses with our microscopes are like this, the convex lenses, all right? So light going, again, from the bottom of the microscope up through the stage to the slide, as we get into the lenses of the microscope, the light hits them and then brings it into a focal point. So a point where we can visualize that, and it's depends on the respective lens where that focal point is at, all right? But you get, like again, flipping it back to that image of the bacillus, a nice crisp image when you are at that focal point. With the concave lens, it's really the opposite, and the focal point is before it moves through that lens, and then we see the light being distorted as it moves through, or you being distorted in the funhouse mirror. So the visible spectrum that I mentioned before is a very small fraction of the electromagnetic spectrum, just a little teeny piece of all of it, all right? All of this energy, from very low energy to high energy. And the waves, again, would be super close together at the high energy end of it, or very far apart at the lower energy. And then moving in to what we can see kind of in the middle-ish, all right, is the visible spectrum. And we can see as we move through here from lower energy right after the infrared region, we get into our reds that we can start seeing, and we move through our rainbow to the violets, and then to the ultraviolet that we can't see, right? So farther spaced wavelengths and tighter wavelengths, higher energy in the blue region, lower in the red, right? And that's what we can perceive, and we can see, you know, respective frequencies and wavelengths, all of that. We don't need to be worried about that. So I mentioned some fluorescence stuff as we were starting this, and this image, I think it's just awesome to see some of these things. We've used a couple of different types of stains that will fluoresce when they are under the right type of microscope, a fluorescence microscope, not just a regular light microscope, all right? It's UV light that excites these pigments in the specific dyes, and it's sort of glow in the dark type of stuff, right? These microscopes are very sensitive, light sensitive. You couldn't, you know, in the micro lab, you got a bunch of benches, you're going to get all of the microscopes out, the lights will be on, you'll be looking through stuff, all fine and good. But to do these things, it really needs to be dark as you can get it, pretty much, right? There is a room in the biology building, the microscopy suite, where there's a bunch of different microscopes, but one room that's really kind of like a little closet. And this closet is where the fluorescence microscopes are. You go in there and you can turn the light on, you can get everything set up, but when you're ready to turn the microscope on, you shut the light off, and now you're in a very dark room, and the stuff starts glowing, all right? Whatever it might be, all right? So here we're looking at some stuff from a cow, bovine pulmonary artery endothelial cells. We've got a lot of stuff in here, all right? So the nuclei, the big bluish purple, they're stained with DAPI. The microtubules are marked in some green with this FITSI stain, and then the actin filaments with this other one, TRIPSI, that give us sort of the reddish color with that. Different structures or different parts of the cell picking up these stains and allowing us to visualize stuff. There's other things where we might stain bacteria, and it might be staining parts of the cell wall or parts, other parts of that bacterium that can allow you some kind of multiple colored things as well, all right? But when we really think about like looking at eukaryotic cells, to me, that's when it really gets impressive, because you can differentiate real quickly what sorts of stuff you're looking at, all right, as we move through here. You could also potentially, witness cells in the process of mitosis, where the nuclear membrane, you could watch a handful of these cells, some of them with the nucleus intact, and then where it's gone away, and then potentially staining up DNA with different stains that allow you to visualize that stuff, all right? Which I think is pretty impressive. All right, so we got to talk about Leeuwenhoek. Antoine van Leeuwenhoek, he's credited as the first person to observe microbes, all right? He looked at all sorts of stuff, all right? He saw bacteria, which to me is really pretty impressive, thinking about the type of microscope that he was building, which is this middle frame, all right? This doesn't look like a microscope to me, all right? This looks really odd, all right? And in turn, he built hundreds of microscopes, ground lenses for hundreds of microscopes, because back then, it's not like what we think about today, and you prepare a slide and you put your stuff on there and you look at it, and then you move it and put a new slide on, and you just keep doing it, right? Each sample that he wanted to visualize, he built a microscope for it, and in turn, kept them on his shelves so he could go back and look at these things, right? Impressive, right? He saw all sorts of things, right? And they think the resolution on this was about 400 times magnification. So bacteria would be visible, but really pretty little, all right? But he could see them, all right? Like I said, all sorts of things, lots of microscopes, right? He's, you know, these little animalcules, animals, molecules, what the heck are they, right? Something, right? They're moving around, swimming around. He saw lots of protozoal organisms, little flagellates moving around real rapidly. He looked at pond water and rainwater, scraped his teeth and looked at the plaque and saw lots of things, bacteria and protozoal organisms. He had Giardia, which is a pretty nasty parasitic infection, gives you some bad diarrhea. So he looked at that too. Why am I crapping myself to death? I don't know. Let's look at it, right? He looked at everything, right? Any sort of bodily secretion, he wanted to see it, all right? Looked at all sorts of stuff, all right? So makes you think this guy must have been a microbiologist, must have been a scientist, but he wasn't. He was a merchant. He was a shopkeeper. He sold fine linens. And in turn, that's really why this first bit of microscopy comes into play. He wanted to know, am I getting quality fabrics like they're telling me, or are you pulling the wool over my eyes, right? That's what he wanted to know, right? Like thread counts of sheets, right? You can kind of tell, right? You could see him with these lenses, right? So he's looking at all sorts of things like these fibers, right? And as he gets these images and starts seeing these things, he starts wondering, geez, what else could I look at, right? And that's where it all kind of opened up as he starts, you know, looking at pond water and other things and sees all sorts of crazy stuff. You see the water and it looks clear, but you put it under the lens and there's all sorts of stuff swimming around. And he's like, what? Nuts, all right? So he's not truly a scientist or a microbiologist, but he opened up the doors for all sorts of things, right? He in turn writes about what he sees, draws brilliant images, draws these pictures of things that he is seeing, and he submits these things to the Royal Society, and people's minds are blown when they're seeing these things. What is it, right? This invisible world that we're just newly seeing. The Queen comes and wants to see all of these things, right? And there were a few things that he shared, and there were some others that he didn't, right? Kind of makes me think maybe some of his bodily secretions. He's like, I can't show the Queen my semen, right? I kind of think that was something that was a no-no, right? But maybe not. Maybe that's what she wanted to see. I don't know, right? I'm curious, right? So some of these he also called wee little beasties, these monsters. I probably think the protozoal organisms that were gigantic swimming around were probably these wee little beasties, right? And the bacteria that are considerably smaller, maybe the animalcules, right? So likely, all right? So another hit here for the journey to the microcosmos that gives some details about this, but I'm not going to click on that. All right. So again, this a replica of some of what he was building. And one thing I skipped over, I want to bring it back to this real quick. As we look at these lenses, all right, and he, as he's building these microscopes, he is making the lenses for these microscopes by grinding this little piece of glass, making it nice and concave, right? And as he's doing this, convex, as he's doing this and grinding it, he's getting very good images, very high resolution, right? And I have read stuff talking about some of this, that it took a super long time for people to replicate the quality of lenses that he was grinding in his little shop. They couldn't compete. They couldn't make them as nice as he was doing, right? Almost a century before they were able to get lenses as high of a quality as Leeuwenhoek was making. And it makes me stop and say, obviously he was a time traveler. He must have been, right? It makes sense, right? He's doing all sorts of things. It kind of makes, there's all sorts of thoughts like that. If you could travel back in time, what would you take with you, right? What would you bring, right? Stink a microscope, right? That's what I want to bring. All right. Crazy. All right. Other way around. Galileo Galilei also pioneering some microscopy, but developing the telescope. All right. And so with that, we think about, seeing stuff super far away that looks very tiny and blowing it up with telescopes, or the other way around, right? And using microscopes to see things right here that are basically invisible. Pretty impressive, all right? Kind of right around the same time. So Leeuwenhoek seeing things, all right? Robert Hooke, similar but completely different, right? He uses a microscope that's definitely a little bit more advanced, right? 1660s with this, and as we flip it back to here, right? Middle of this, right? We've advanced a bit. Still pretty primitive looking. A candle for our light source, mirrors to focus in on this, and then some lenses within here, and you'd look through this little ocular at the top and see things, all right? He looked at lots of things as well, but we credit Robert Hooke with coining the term cell in the first place. He looked at cork cells and a special type of oak tree, the cork oak, gives us the bark within there, super thick, gives us cork, all right? And he made very thin slices of this bark, the cork, and put it under the microscope and could see the cell walls of this cork. And these little kind of rectangular shaped cells, as he looked at them, he thought, these look like little rooms. They remind me of the rooms that monks live in the monastery, and those are called cellulae. And that, in turn, is what he called them, the cells of the monastery, right? The cellulae. So, right around the same time, we start seeing all sorts of things and start talking about cells, and there we go, all right? Another time traveler, maybe. Zacharias Janssen and his father Hans could have invented the telescope, the simple microscope, and compound microscope during the late 1500s or early 1600s. Interesting for sure, right? And definitely right around the time of Leeuwenhoek, as he's grinding his lenses and seeing these things, there are others that are truly trying to visualize things that are difficult to perceive with the naked eye, right? I think this is pretty impressive because Janssen is still a name that you can see with numerous devices, telescopes and microscopes for sure. some still bear their names, right? And I think that's pretty cool, all right? They've been in business for centuries, right? Impressive. All right, so this image is a quick anatomy lesson of the compound microscope. You're going to see microscopes that look pretty similar to this in the lab, all right? There are all sorts of pieces in here. We'll go through and talk about it quickly. I know in the lab, when we get close to the microscopy stuff, your TA will go through and talk about things and show you stuff, how to properly deal with it and handle it and clean it and put it away, all of that stuff. But I'll give you a quick spiel so that you're at least a little bit familiarized with it before you get in there, all right? This looks a heck of a lot different than Leeuwenhoek's microscope, right? And like I said, we take some of this for granted because we put slides on the stage, move them off, put another one on, and just go about our business, all right? So there are lots of bits and pieces. These microscopes are fragile. You want to be very careful when you're getting them out of the cabinet and moving them around and putting them back, carrying them with two hands. They're all about $2,500 a piece or so, kind of expensive, right? So we want to be careful with them, right? Starting off at number one, I suppose, because that's probably where you ought to start. The eyepiece up at the top. This type of microscope is what we call a binocular microscope because there are two ocular lenses or eyepieces, right? The one with hook before was a monocular, a single lens, or like a telescope, right? With the single lens that you're looking through. So you're using both of your eyes to look through there and focusing in, right? You should be using both of your eyes, right? You can adjust these oculars. If your eyes are closer or farther apart, you can move it a little bit, right? You can also turn the knobs up at the top. That helps you maybe potentially focus stuff in. Once you have done that, if you look at, there should be a marker on there. It says, you know, you've turned it, looks all fine and good, and it says 6. Any other microscope you get, you can do the same thing and just turn it to 6. And you should be good to go, right? If you wear glasses, because I look right here, if you wear glasses, you technically are supposed to take your glasses off as you're looking through the microscope. I've seen people going both, right? Kind of just depends. And I don't wear glasses, so I can't say one way or the other, right? But that's also the little knob up at the top is supposed to allow you to dial that in so you can see stuff without your glasses, right? I also see lots of people looking through there with one eye, like a pirate, all right? You really should be looking at with both eyes. And if you're having trouble, just try manipulating some of that, and that should help. If you look in there and you see lots of little black hairs, you're seeing your eyelashes. You're too close to those lenses, and you need to just move back the littlest bit, all right? And there's other details. If it's not a full circle, that might be something down below, like this diaphragm, where it's not completely put in there. It also might be that the objective lenses aren't completely clicked into place, and that doesn't give you a complete circle. It gives you a partial, a half moon, or something like that, where you're not getting it all, right? So you look through the oculars, it goes down and to the objective lenses, and there is this revolving nose piece, because most of these microscopes have a handful of lenses on them, all right? 4 different lenses from low power to high power. When we first start looking at stuff, we look at the low power, and then we go to each respective lens till we get it to high power, all right? When I want to look at bacteria under the microscope, these suckers are super little, all right? So I get everything set up after I've stained everything and all of that stuff. I put my slide on the stage, and I bring my stage, all the way up to the top, all right? I use the coarse focusing knob, the bigger knob, and I bring the stage all the way to the top. That way I know all I have to do is slowly go down the littlest bit and I should start seeing stuff. If I start at the bottom, it's going to take me forever to move my way up as I'm staring through these lenses. It's going to take forever to get almost to the top to start seeing stuff, right? Bacteria is small. You need to be toward the top, right? So you start seeing stuff, you're using your course adjustment. And once you start seeing the hint of things, color is probably going to be it. You can fine tune it the littlest bit, all right? The low power is not really going to allow you to see bacteria, but you can see the hints of something there. Then you move to the next lens. These microscopes we call parfocal microscopes. So when you're in focus on the low power and you move to the next lens, you're very close to in focus, and you don't need to use the coarse adjustment, use the fine adjustment knobs, the littler knob, all right? And I can't tell you because it truly kind of varies. You either move it away from you or towards you, just the littlest bit, a small little turn, slow, all right? And what you saw on the first one, you should see a little bit better on the next one. And it should be pretty quick to kind of get the hint of some color or something like that. Once you got that, you move it to the next one, a little more fine tuning. Now you're probably at this point at the 400 magnification, you're going to start seeing bacteria. And then a drop of oil as you move the lens out of the way, drop of oil onto the slide, moving the oil lens into it, and another quick fine tuning. And it should look good. You should see the colors and the cells. You can get all sorts of details, but again, they still look pretty small, right? The oil and the objective lens need to touch. And if you've got your objective lens way up above that slide, it's not touching the oil, you're not seeing the right stuff, all right? It's not happening, all right? So there is another knob at the bottom of the stage that moves the slide left and right and up and down, back and forth, whatever. So you can move your sample around and look at that. You can adjust the amount of light moving through there with the diaphragm. The condenser focuses light in. There's a knob over here that turns the power on typically. Some microscopes have a switch to toggle it on and a knob that adjusts the light intensity. Most of them, it's the same thing. You turn the light on, and with the lowest power, you should have barely any light moving through that microscope, pretty dim, because it's going to be a big field of view that you're looking at. And if it's all the way on full bright, you're going to get a headache real quick with the amount of light that's moving through there, right? Really kind of dim. As you get to the highest magnification, you're going to turn the light up all of the way. You want as much light moving through there so that you can really see these samples, whatever they are, nice and crisp, right? That's the idea. So the base of the microscope and the arm back here are what you're going to use to hold it, right? To carry it to and from your bench. And I think that was a good point to stop. Good day to all.